Sometimes colleagues accuse me of studying vision, and I always correct them and I say, no, no, no. I use vision to study plasticity in the brain. It was at that moment that I realized, well, if I can do this as an undergraduate, there are probably some more discoveries to be made that maybe I can contribute to brain science, despite the fact that there's a library full of books on the brain. Scientific career is really wonderful because you never know, when you keep turning rocks over to see where they're going to lead you, and you never know where it's going to go.
In this episode of the 632 Nanometer podcast, we sit down with Dr. Mark Baer, a distinguished professor of neuroscience at MIT, whose groundbreaking research has reshaped our understanding of how the brain adapts and changes. Using the visual system as a window into brain plasticity, Dr. Baer's work has uncovered fundamental mechanisms of how our experiences modify neural connections. His pioneering studies on amblyopia, commonly known as lazy eye, have not only advanced our understanding of this condition, but have also revealed crucial insights into how the brain processes visual information during both waking hours and sleep. Through careful experimentation with mouse models, Dr. Baer's lab has made remarkable discoveries that extend far beyond vision, offering new hope for treating conditions like Fragile X syndrome and autism. Join us as we explore the fascinating intersection of vision, sleep, and brain plasticity with one of neuroscience's most innovative minds.
Cool. Since you have been studying brain over a long time, so what's your favorite part of the brain?
My favorite part of the brain is the cerebral cortex. And my favorite part of the cerebral cortex is the visual cortex.
Why visual?
Why visual? Well, there are a couple of good reasons. One is about 50% of the human brain is devoted to the analysis of the visual world. So I'm among many thousands of scientists with an interest in vision, hardly unique. But for my personal interest is the deeper question that interests me is how experience modifies the brain. And the visual system is very useful to study that question because it's a relatively simple matter to manipulate visual experience and measure the consequences.
What was the time when you realized that a visual cortex or vision system is of interest to study? How did you get there?
Oh boy. Okay, so you want some deep background? I'm going to give it to you.
Yeah, let's start from there.
Like many people interested in the brain, I had a medical condition that interested me in what's going on under the hood. And the condition is called congenital nystagmus. So my eyes move back and forth involuntarily. It's an exaggeration of the normal eye movements that we all have called saccadic eye movements. But those of us with congenital nystagmus, our eyes go back and forth. And I just want you to know the world is not moving for me, even if you see my eyes moving. Interesting question in its own right, but vision is stable, but it's degraded. So I have impaired vision.
Is it literally blurred because there are not enough frames being captured?
I think that's a good way of thinking about it. Yeah. It's measured as my best corrected visual acuity is not 20/20. It's more of an annoyance than a serious disability. I couldn't qualify for military service because of it, so I'm grateful for that. Anyway, so, I have this condition, and when I was in college, I wanted to learn more. And at that time, neuroscience was really still in its infancy. In fact, it wasn't even named neuroscience at that time. So really the way to get there either was very often it was through psychology, which was the closest discipline that was informing people about the brain function, especially physiological psychology, it's called. I'm the beneficiary of an excellent liberal arts education. I went to Duke, and I was exploring a lot of different subjects along the way. And I considered a lot of different career options along the way. But I think for me, the transformational event was when I was offered the opportunity to work in a lab. And this is working on the visual cortex. These are the early days of tracing connections in the brain. So you would inject a marker into the brain, and then you would allow for a couple of days for it to be transported, and then you would reveal where the marker went. And we were just working out the earliest crude wiring diagram of the brain beyond what was possible with a crude dissection. And there was this suspicion that there was an input to the visual cortex from a part of the forebrain, an obscure structure, it's so very mysterious, called the claustrum. As an undergraduate, I was simply following instructions, and I was told to inject a tracer into the claustrum, find it, inject a tracer into the claustrum, and trace the connections. In those days, it took about a month to to actually reveal the connections because it had to do with exposing a photographic film to radioactivity. So it took a long time to actually see the outcome of your experiment. You talk about delayed gratification, right?
Hmm.
I remember the day when we first developed the slides to look at where these what was going on with this connection. Of course, I'm a young student. I have nothing to compare this result with, but the postdoc that I was working with was like jumping up and down and they were ready to break out the champagne. And we had discovered a connection that no one had actually recognized previously. It's a major connection. And it was at that moment that I realized, well, if I can do this as an undergraduate, there are probably some more discoveries to be made that maybe I can contribute to brain science, despite the fact that there's a library full of books on the brain. Then I was deeply immersed in the visual system and the visual cortex. I just loved it. I love the structure. And we were also leveraging at the time the excitement around the early discoveries of Hubel and Wiesel, who were working at Harvard Medical School at the time. And they had been mapping out the organization of the ascending visual pathways with special emphasis on the visual cortex. So more was known about the visual cortex than any other part of the cortex, for sure, thanks to their work. So it was just a coincidence that I was immersed in the visual system. The most exciting work at that time was coming out of studies of the visual system. This work was attracting what you might call the best and brightest in the field to work on vision and visual system. And then when I went off to graduate school, my emphasis shifted from the visual system to really plasticity of the visual system. And this, again, Hubel and Wiesel were there first. They brought attention to this fact that you could do a simple manipulations of visual experience and really profoundly modify the organization of the visual cortex. From then on, I was hooked, and I've hardly wavered from that early influence.
Maybe if we can go into deeper understanding of how like the vision works and vision processing and how the plasticity involved in this process, if you could give a brief introduction into that.
Sure. Yeah. I mean, obviously, it's a vast topic, right? So this will be a thumbnail sketch, but I'll get you to what interests me. I should preface this by saying that sometimes colleagues accuse me of studying vision, and I always correct them and I say, no. I use vision to study plasticity in the brain. That's a bit of a cop-out because that means I don't have to understand in depth the intricacies of visual processing. So just to answer your question, obviously vision begins in the eyes. So much of the eye is devoted to forming a crisp image of the world onto the retina. And light is detected in the retinas. There's a lot of pre-processing that's occurring in the retina. And then ultimately, this culminates in nerve impulses, action potentials that are generated by retinal ganglion cells. So those are the major output cells of the retina. So the ganglion cells send axons that go down the optic nerves. There's a partial decussation at the optic chiasm. That means some of the axons cross, some of them stay on the same side. And it's that judicious crossing of information from one side to the other that gives rise to the organization so that the left hemisphere of the brain is concerned with the right visual field and the right hemisphere of the brain is concerned with the left visual field. So those inputs go in, they're en route. They have a number of targets. So some targets are related to the control of eye movements. Some targets are related to the control of circadian rhythms. But the major target that is responsible for conscious vision is a collection of neurons in the thalamus of the brain called the lateral geniculate nucleus. That's the first synapse in this direct pathway of visual consciousness. The lateral geniculate is an interesting structure in the sense that it keeps the inputs from the two eyes still strictly segregated. This is very clear in primates and other species that have excellent binocular vision. So there's a layer of neurons devoted to each eye, and that can repeat several times depending on the species. In fact, when Hubel and Wiesel first explored the organization of the lateral geniculate nucleus, they were unimpressed. They thought it seemed very much like the retina. So they were said, not much going on here, and on to the cortex. So then the lateral geniculate nucleus projects axons into the cortex, and there they saw an elaboration of the type of stimuli that drive vigorous responses in the cortex. And from my point of view, the most remarkable discovery was simply that individual neurons in the cortex now responded to stimulation of either eye. So there was this very precise mapping of the inputs from the two eyes onto common cortical targets. So I like to say this is the neural substrate of binocular vision. That's why we see one world through two eyes. And I should just add as a postscript, because I get in trouble depending on who's listening to this, that the lateral geniculate nucleus turns out to be very interesting. Still rather mysterious, at least to me, but the major input to the lateral geniculate nucleus is not the retina, but actually it's the cortex itself. So it's a first site in the visual pathway where there's some very important feedback regulation of what's going on.
I see. I'm more, I think, familiar with, let's say, like neural networks, which do with processing of images. I'm curious, how is it close to what it happens in the eye? So, for example, the convolutions, I learned that it happens with rods and conuses, right? With the colors.
Yes.
And so some of the convolution happens there. And where like other things, let's say segmentation or semantic analysis of what we see, let's say I see now this like the table. So how do I know where are the edges or at which distance each of the objects is located? And how does it work?
I think you're luring me into a discussion of vision,
Okay.
which I just want to remind you, I don't study vision. I use vision to study plasticity.
Okay.
but I'm happy to opine. So yes, as you said, there are two broad classes of photoreceptors in the retina. One, the rods and the cones. The cones are interesting for a number of reasons. One is they're very numerous in the regions of the retina that serve central vision. So they're really responsible for high resolution vision. The rods are much more common in the peripheral retina. That's the surrounding region. The rods are more interested in overall luminance and perhaps moving stimuli. The cones are very interested in fine detail. There are three types of cones that are distinguished based on their wavelength sensitivity. So by ratioing the output of these three types of cones is essentially how we detect the full rainbow of different wavelengths of light, of visible light. It gives us all the colors. It's just a simple ratio of these three
Is there a
cones.
mathematical reason that it's 3, just like cameras? Could it have been 4 or 5
That's
or 2?
a super interesting question. I don't have a good authoritative answer. I'd say, well, 3 is obviously the minimum. We couldn't get by with 2. In fact, there are a lot of people with red-green color blindness that have impaired, they sort of do scrape by with less than the optimal number. I'm also colorblind. But it's interesting you should say about 4, there is no reason why 4 couldn't be better. In fact, there are people that are considered to be quadranopes, an additional type of photoreceptor with a slightly different wavelength sensitivity. So they can discriminate colors that we can't, like shades of white, for example, that are really hard for some of us. Yeah. So anyway, so the photoreceptors are the business end, right? They absorb photons and they turn that into voltage. The voltages modulate neurotransmitter that's released by the photoreceptors. And then there's some pretty elaborate processing that's going on in the retina so that you already have a well-processed output by the time you get to the ganglion cells.
When you say processing, what do you mean exactly? Is there some integration going on or something like that?
Yeah, there are lateral interactions. What you see immediately is different channels of information. So there's a channel that's sensitive to light. There's a channel sensitive to dark. They're wavelength sensitive channels. And a way of exploring this is to sort of work backwards and start at the level of ganglion cells. So those, like I said, they're the output cells. And you can ask, what are the preferred stimuli for ganglion cells? And what you'll find is that the cardinal feature of ganglion cells is what we call receptive fields. Okay, that's a general concept in neuroscience. A receptive field is sort of that part of the visual world that elicits a response in a particular visual system cell. Okay, similar sense of touch, different parts of the skin elicit a response to different neurons, right? That's a receptive field. And the receptive fields of the ganglion cells are interesting. They're usually divided into two regions. They're like concentric circles, and there's a center region that'll either respond to a light on in that region or a light off in that region. So you already immediately have two separate channels, on and off. The surrounding region is antagonistic. So as a stimulus starts to encroach, like a light stimulus in an on-center ganglion cell, as that light starts to encroach on the surrounding region, then the response diminishes. So they really seem designed to detect contrast borders, not absolute levels of illumination.
So how do we get to the plasticity in the story?
Oh, thank you. I'm so relieved to hear that question. it's been a long and really fun journey for me, my studies of plasticity, but it really begins with, again, Hubel Wiesel, David Hubel and Torsten Wiesel. And they, or at least the way I tell the story, I think this is more or less accurate, is in pondering the precision required to have the precise mapping of the inputs from the two eyes onto common target neurons in the cortex. That is to say, let me just go back to the receptive field concept for a second. So when I talk about a ganglion cell receptive field, it's going to, that ganglion cell will respond to stimulation only of that eye, right? If it's a right eye ganglion cell, you're not going to get a response by stimulating the left eye. You're only going to get a response by stimulating the right eye. When you go to the lateral geniculate nucleus, same story. You've got neurons that have receptive fields. And in fact, those receptive fields do resemble retinal ganglion cell receptive fields. But again, typically, those cells will respond only to stimulation of one eye or the other eye, never to both. When you get to the cortex, then the miracle occurs that you find individual neurons that respond to stimulation both eyes at precisely the right location in those eyes, right? So there's an appropriate mapping of the visual field as imaged in the two eyes onto a common target cell. So I think that that degree of precision seemed to be greater than what could be achieved with genetic instructions alone. That was the thinking. Now, maybe they were underestimating the power of genetics, but the truth is that there are a lot of other mishaps that occur with the physical development of the body and the head and where the eyes are positioned. And so it kind of makes intuitive sense that there would be mechanisms in place to fine-tune those connections based on visual experience, based now on postnatal visual experience, to tune up those connections and to
What you're saying is that there's one cell that's responsible, let's say, for some pixel in the visual field, and it needs to be tuned to exactly which pixel in the right eye and which pixel in the left eye are talking to it so that it can be stitched together.
that's exactly.
So, like, if I
right.
obstruct one eye, I still see the same thing, in my
That's
final
right.
vision. Okay.
That's exactly
And
right.
there's small chance there would be no error in this. So you have to, as a baby, bring these images together somehow by adjusting the connection.
That's right. That's right. And those connections also, of course serve binocular vision in the sense that we see one world through two eyes, but also those connections serve stereopsis. That is the ability to detect depth. So it's critically important, the survival of species that require good stereoscopic depth perception, like a bat swooping down to catch a mouse. If they crashed into bat's a good example. Come to think of it, let's go to an owl. An owl swooping down to catch a mouse, miscalculation, and they would crash into the ground. Right. So that was the thinking, was that this fine-tuning process very likely involves visual experience, kind of learning. And the test of the hypothesis was a simple experiment as you can do in neuroscience, I think, which is that they close the eye in young animals. They could do it by suturing the eyelids closed. They could put a contact lens on one eye that blurs the image. In the early experiments, they could do with this manipulation we call monocular deprivation. If they did that for a week or 2 weeks or a month or so, and then they restored vision in that eye, what they discovered when they then went to Cortex and recorded the activity is that the neurons responded only to stimulation of the other eye, that we call in the field the fellow eye, the non-deprived eye. So it loses all the connections to the deprived eye.
And can these connections be restored?
They can be. And that's there's so many exciting questions that come out of this work, right? So the question is, well, how do you lose the connections? How do you restore the connections? And one thing I will say is that both losing the connection and restoring the connection is possible, but only during a finite period of early postnatal life. And that is what we call a critical period. Okay. There are critical periods of many facets of brain development. This is an example in the visual system, the best studied example.
And how long is this period lasts?
It depends. Yeah. In humans, in a way, it has to be operationally defined as the period of time when a certain manipulation can produce a certain effect. Okay. If that makes, I'm being a little cagey here. If you were to recreate in a human the conditions that Hubel and Wiesel used they were originally working in cats and also in monkeys, that critical period ends before the first year of life. So it's a very early critical period. If you do more subtle manipulations of visual experience, you can still get very robust effects, but then those can be extended out to 7 or 8 years of life. So it's so loosely defined. Typically, if you read about it, the claim will be that the critical period ends at around age 7 in a human.
And what kind of processes define this critical period?
So let me be a little bit more explicit about what I'm talking about too. There are two ways to probe a critical period. I mean, these are the ways we do it in animal models. One is to define the age range at which commencing with monocular deprivation changes the responses in cortex. Does that make sense? So in other words, if I were to deprive an animal of vision in one eye for a week, and I started that at different ages, I could map out what age does that manipulation no longer have any effect. So that is one definition of the critical period. So that would be, to be really precise, it's the period of vulnerability to deprivation. The flip side is you've monocularly deprived and then you've reopened the eye and done a manipulation to promote recovery. How long is that possible? At what age is that no longer possible? Now, fortunately, both of these measures of so-called plasticity overlap to a great degree. But I think it is also important to be precise in how you're measuring the critical period because the rules need not be the same for vulnerability and recovery. So I think your deep question, and the deepest question of all, I suppose, is what brings the critical period to a close? Is that your question?
That's right. Exactly.
Well, this has been a question that has challenged a couple generations of neuroscientists. And I don't think there's going to be a single simple answer. I can tell you what's the prevailing theory, which is the prevailing theory is that inhibition in the brain is late to mature. And when it achieves a certain level of maturation, that ends the critical period. And this hypothesis is well grounded with data. I think what's less clear is what is it about inhibition that's, constraining the type of plasticity that's present. So I think there's still some questions, next level of questions that are really important to address, but you can sort of break it down in a way. The way of thinking about it is there are patterns of activity that are arriving in the cortex. For instance, in the case of monocular deprivation, you have well-correlated patterns so neighboring ganglion cells are active at the same time when you're looking at the visual world, right? You're seeing shadows cross the retina, neighboring cells are active at the same time. We call that correlated activity, right? The other eye, an eye that's been deprived, that we would call that noise. It's not no activity. The retinas are quite active in the dark. Retinas are active unless we do something active to get rid of that activity. But it's patterned activity we replace with noise. So there's a process in the brain that is making a judgment about whether a synapse should be strengthened or weakened depending on whether it's a patterned input or a noisy input. At a basic level, that's the way it's got to work, right? So that is a very robust process during early postnatal life, and then it fades with age. So how come? So one possibility is that the molecular mechanisms of the synaptic plasticity that we'll undoubtedly talk about change with age. So, proteins that are expressed early in development are no longer expressed in adults, and that just the whole system sort of freezes out. That's one possibility. Another possibility is that these patterns of activity are no longer gaining access to the modifiable synapses. And that's how I view the role of inhibition, because inhibition can constrain the relay of activity in the cortex from one layer to the next and so on. And if that activity is not making it to the modifiable synapses, it's not going to have any impact on those modifiable synapses. Another hypothesis that has been around for a long, long time is modulatory inputs to the cortex, the inputs that convey information about salience and whether these modulatory systems are dialed down with increasing age. So a lot of very interesting hypotheses have been floated around for a long time. I think they're all going to be, in a sense, correct, right? So I don't think any have been excluded. But probably the one that gets that's the textbook version right now is something to do with late maturation of inhibition. So let me say a word about that. So brain development occurs in waves, and very early in development, there's a creation of excitatory synapses. For instance, we talked about lateral geniculate nucleus and cortex. So those LGN neurons give rise to axons. They form excitatory synapses onto cortical neurons, and that's how we see. And there's a great deal of synaptic proliferation early in utero and after birth. It goes crazy just to get the system wired up.
How do people know this? Like, how do you actually study this? Do you have to freeze dry fetuses and slice them into little slices and look in a microscope? Or like, how do you know that this is happening?
All these studies have been done. Yeah. Anatomical studies, electron micrographic analysis, looking at synaptic density. One of the most amazing.
Is it something you can study on a live animal, or it has to be done by like actually
you
slicing?
can nowadays.
Okay.
It's a question of access and how long you can image. But nowadays it's amazing what can be done. So you can actually watch synapses as they're formed and follow them for a long time. In fact, I have colleagues right down the hall that are doing on a developmental time scale. That is, we're talking about months. That's a little harder. But a fun fact is that there's a overproduction of excitatory synapses in the visual cortex. So they will peak at about 150% of the adult value in early childhood. And then there is a mad process where those synapses are whittled away during adolescence. So you lose about 50% of those synapses during adolescence as you're fine-tuning your connections.
So it's interesting. Fine-tuning or learning math in school is just about pruning.
Yeah, sure. Sounds good. Yeah, that's it. Problem solved. Okay.
Cut down the rest.
That's right. The more synapses you lose, the smarter you are. Quote me on that. So we've just been talking about excitatory synapses, but there is, in addition, everything you get in the brain is a balance of excitation and inhibition. And in fact, crudely, we always talk about what's called the EI ratio, the ratio of excitation to inhibition. So the EI ratio favors E early in development, but there is the late arrival of inhibitory neurons. The neurons themselves have a different migratory path to get to the cortex as it's developing, and the connections are slower to form. So the full maturation of inhibition coincides with roughly the time when that synaptic pruning stops. So these are conspicuous coincidences that inhibition matures at about the time you lose plasticity. At least in mice, it's possible to manipulate that development of inhibition, and you can manipulate the duration of the critical period in some fairly predictable ways. So that's what's really given the most support for this hypothesis.
So you mentioned that in your experiments, one eye is the fellow eye, like the working one, and the other one is the lazy one. So what happens for people who have both impaired eyes or maybe even like blind person, how the brain develops?
Well, I think one of the things that impresses me is that you think about, say, monocular deprivation as being a deleterious effect because you're losing responses to the deprived eye. Looked at it another way, the deprived eye is not really conveying any useful information. You're not forming crisp images on the retina. It's just a bunch of noise. And what you're really doing is taking maximal advantage of the neural hardware that you've got by decreasing the weight of that input and increasing the weight of the input from the fellow eye. So it's a compensation.
Mm-hmm.
And in fact, anatomically, this is very dramatic in the human visual system and the monkey visual system, where what you see is the amount of brain tissue that's devoted to the analysis of the eye that had been deprived shrinks quite dramatically. And you get a compensatory increase in the brain tissue that's devoted to the analysis of the other eye. So no brain is going to waste. And this is also true with congenital blindness. Like the regions of the visual system that normally serve vision are now being used to serve other sensory modalities. So the brain doesn't go to waste.
So something else takes the
yes,
the usage
it is an area of neuroscience that I'm not following closely, but there are a lot of examples of enhanced, and to give an example, Braille, for instance, the ability for blind people to read Braille. I mean, that is a very fine tactile skill that is very difficult to achieve for a normally sighted person. And yet it can be done because you're really taking full advantage of the cortex that otherwise would be devoted to the analysis of the visual world.
Also, what about when the eyes are closed? I guess we're maybe going towards the area of the sleep. So one idea is, I understand that we get information from the eyes, then it propagates to the brain, but then we shut down the channel, and then let's say when we close our eyes, so what we like, for example, imagine, or it could be maybe by stimulated by some drugs like psilocybin, something else. So what happens in the brain at this moment? Like, how do we see with closed eyes in this case? I mean, I guess I mentioned like several different directions, but what happens in this case?
That's such an interesting question. I guess there are a number of layers to this, and don't let me forget the question as I ramble on, okay? But one of the questions is, if you take a young animal or person and they're visually deprived through both eyes, that's a condition called binocular deprivation, and that will also degrade vision. It's not as dramatic as what happens when only one eye is deprived. It's much more dramatic, but nevertheless, it'll still be a degradation of vision. The neurons will lose responses to the stimulation of both eyes. And so we do that experimentally. We do binocular deprivation. We close both eyes, and we have these effects that we can measure. And then you can ask yourself, well, normally we spend about 50% of our life binocularly deprived because, just as you pointed out, we're asleep. Our eyes are closed. What's going on? And I think that was one of the many suggestions that maybe these neuromodulatory inputs to the cortex are critical for the plasticity. So we haven't talked about neuromodulation, so I got to introduce it. But we've been talking about some very precise mapping of retina onto lateral geniculate nucleus onto visual cortex, okay, point-to-point communication. But there are other systems that we call diffuse modulatory systems that are responsible for general levels of arousal, alertness, and their activity changes a lot during a sleep-wake cycle. So a lot of the modulatory inputs that are active right now, as we have this conversation, will be shut off entirely tonight when we're asleep. So that was one of the early suggestions, that maybe these modulatory inputs are responsible, or necessary anyway, for a gain control mechanism on plasticity. So they're just shut down at night. So everything freezes. Don't worry, your synapses will still be there tomorrow when you wake up, right? And of course, we have our own subjective experiences in our mature visual systems of visual imagery and things during dreaming and so on. Usually it's a bunch of nonsense stuff that you can't possibly describe the next day, but it seems vivid at the time. Now, there are hallucinogenic states. They can be created by hallucinogenic drugs, as you mentioned. And I think that an important concept is that vision does not depend on the eyes. Vision depends on the brain, or the rest of the brain. In other words, I could take your eyes out and I could stimulate neurons in your brain, and you would think you were seeing visual stimuli, right? I mean, it's the activity of the brain that is registering conscious appreciation of what we call vision. Normally, we do rely on the eyes, but that's the story. In hallucinogenic states, the activity that sweeps across the visual cortex is actually not generated by the eyes. It's generated by connections within the brain. And therefore, you can see, quote unquote, see things that you could never see in life, combinations of colors and things that I should preface this by saying I've never taken a hallucinogenic drug. But what I read suggests that, anyway, that's the kind of synesthesia, merging of different senses, hearing colors and seeing sounds and things like that.
That's one aspect. Maybe people don't talk about it as much because it's not as surprising. But the more typical hallucinations from psychedelics are like, people look at their hands. That's a very classic one where people are staring at their hand. Why are they staring at their hands? Because they're changing size a little bit. So they're like breathing a little bit.
Yes.
And that seems more like maybe just noise in the system. It's not like a new image, right? It's just your hand. You're looking at, is it getting a little bit bigger, a little bit smaller? It's like, whoa, wait a minute. Like,
draws your attention.
Yeah, it's like, it's strange. What size is this actually, right? So that's feels more like maybe not something new or synesthesia or something, but maybe something is misfiring in the chain that's trying to figure out.
I think that makes sense, right? That's exactly makes sense, is that the constraints on vision are, some constraints are removed. You get a wobbling perception. One trigger for hallucinations actually is just going in sensory deprivation, or even just complete darkness. So if you're put in complete darkness for a period of time, patients will develop visual hallucinations. So the visual system is making up a story that doesn't require the retinas, right?
So this concept of plasticity, there's a lot of at least pop science stuff about plasticity, right? like psychedelics or other experience, like, oh, this increases plasticity. There's kind of like this pop science use of the word plasticity. How does that actually relate to the hard science of plasticity? Like when they say psychedelics increase plasticity, or, I mean, one thing that I've read about, which I thought was very interesting, is they would, in like the Middle Ages, they would take children to executions in order to scare them, right? Or they would even, I think this is in Ireland, in order to sign a contract that somebody would remember a long time in the future, they needed a child, because all the people who are signing might die, because it's like a land deed or something, and there's no paper. It's like a, handshake.
Yeah.
So they would beat the child at the day of the contract in order to like make sure that they remember this moment. So it's like an extra plastic day where they like really rewire them to remember.
Ghost alone memory.
Yeah. So the question is like, what's the connection between the like pop science idea of plasticity versus the real world's like down to earth research?
There are some strong connections. There are some, believe it or not. Believe it or not. In fact, we were just talking about that today in my class, right? Is the flashbulb memories. We're getting far afield from vision, but I hadn't heard about the child beating, but and I'm not advocating it. But I don't know if you all want to get into this, but information streams in to all our sensory systems and lays the tracks of memory, and most of those tracks will fade with time. That's normal. It's adaptive, because most of the information that goes in isn't really valuable for our survival, right? On the other hand, there is some information that is critical for survival. So there's systems that sort of weight the information. And one of the sort of variables in the weighting of this is whether you recruit new protein synthesis in the brain. And a way of doing that actually is to turn on these modulatory systems I referred to earlier. The example that, in my generation, or your generation, I hope, was 9/11. Like, where were you when you learned that the Twin Towers came down? It's not that I'm asking you to remember that the Twin Towers came down, because obviously we're reminded of that. It's the context in which you learned that. You remember who you were with, what you were doing. A lot of details that otherwise would have faded away for good reason are now seared into your memory. And so every individual has examples in their lives of these so-called flashbulb memories. So I can see the poor child's like, what the heck are you doing to me? And so, yeah, at that moment, it's a traumatic experience, and it's activating all these modulatory systems. And you're damn right I'm going to remember every detail of what happened.
But is that a plasticity thing? Like you're actually rewiring some neurons in order to make that memory? Yeah,
I would say the plasticity is occurring whether you want it to or not. But what's changing is the persistence of the plasticity in that case. Right. So yeah, where plasticity happens, we'll remember this conversation for a period of time, and that's because we've laid the tracks of memory. Synapses have been modified in the brain, and that's the plasticity. And then these connections may fade, these modifications may fade with time. And the older you are, the more likely that is.
Is there a path to, for example, taking a drug that would ensure that whatever you're doing at that moment, you'll remember?
Yes.
There is,
yes,
because that would be extremely convenient for a lot of things,
It is.
Forgets.
I mean, that would be or forget. Yeah, exactly. If you could choose.
Yeah. Forgetting is probably more therapeutic, but yes, the answer is yes. In fact, there was a big push in I want to say the 1990s, late 90s, early noughts, to develop memory-enhancing pharmaceuticals. It was called the memory space in biopharma. It's like, we got to get into the memory space, right? And develop drugs. And the whole concept was to take a drug that would actually boost the actions of these biochemical pathways that can convert a temporary change into a permanent change. And there are drugs they'll do this. But the problem is that every drug that I'm familiar with has side effects that make it not therapeutically useful. They can still be abused.
What's like an example of the drug and the side effect, and like how does it work?
I don't want to give examples that sound too benign and promote the use of these drugs. You have to look that up. But one of the key steps in converting a temporary memory into a long-lasting memory is the generation of a second messenger in neurons called cyclic AMP. This cyclic AMP turns on a pattern of gene expression that is known to regulate the persistence of synaptic plasticity. So one of the early attempts to develop memory-enhancing pharmaceuticals was to develop drugs that inhibit the enzyme that degrades cyclic AMP. That's an enzyme called phosphodiesterase. And in animal studies, it can be shown that, yes, manipulating phosphodiesterase can really profoundly modify persistence of memory. The problem is that the drugs used in humans cause terrible gastrointestinal effects. So as we joked, it would sear the memory of you sitting on the toilet because it was not a great experience. And they couldn't work their way around the side effects to be viable.
Is that related to like the delivery? So if you take it orally, maybe versus taking it, I don't know, intraspinal?
No, it wasn't. Well, yeah,
something?
maybe if you took it in direct injection into the brain, you might get away with it. But actually, there may be I don't want to call it a happy ending to the story, but there's a specific isoform of this enzyme that was the sort of thought to be maybe the magic bullet because it's expressed highly in the brain and not in the GI system and so on. And it took a long time to develop small molecules that could selectively target that. And I think there has been a breakthrough in that. In fact, they are in clinical trials now.
And so that would be like for dementia, probably, right?
I think that the big objective is develop a drug to help people with dementia, age-related memory impairment, Alzheimer's disease. The studies that they're doing, and maybe we're going to talk about this at some point, actually more advanced studies are done in a rare condition called Fragile X syndrome, which is a form of intellectual disability. There's really a different rationale for using these drugs in Fragile X than the one I just gave you, but I think that it's being used as a test case to assess safety and things like that of the drugs. So we'll see. We'll see what happens.
Does this somehow correlate also to people who have, these almost like pathological photographic memory, like the kind of people that can look at one picture of Manhattan and then draw it from memory?
Yeah, amazing, isn't it? I mean, truly amazing.
Yeah.
And that is a debilitating gain of function, right? Is that you can't get rid of some of this.
But are they
stuff,
biochemically, for example, like is that enzyme broken or like something like, has it actually been figured out, like how they're, what's
I don't
different
know
about
the
these
answer.
people?
I don't know the answer. And I'm not even sure the question has been asked because it really depends on what path you've taken to get to the question. I would say that'd be extremely interesting to understand that, whether there's a clear biomarker of that type of persistent and unusual plasticity.
So the plasticity, as you mentioned, is an ability of brain to modify itself after certain experiences. And in terms of visual cortex, the main experience we've been talking about is to deprive the vision. What other major experiences we can have in life to modify our visual cortex?
Great question. So we've been talking about monocular deprivation because Hubel and Weasel got this game started in 1963. And these experiments are still happening all around the world as a paradigm to study experience-dependent plasticity. And one of the reasons, there were a couple of reasons why this has been pursued so intensively by generations of scientists now. And one is that, it's a question of brain development. It's a question of how synapses form and are modified by activity. So it's a very fundamental aspect of brain development and whether we could gain insights into that. A second reason that it attracted a lot of interest is it actually models a human clinical condition that we may talk about later, but called amblyopia, which is a condition where children are born with unbalanced vision in the two eyes and the brain doesn't wire up correctly, and there's a permanent visual disability unless it's treated early in life. But the third reason that it's attracted a lot of interest is that, at least at the time, it was the first robust demonstration in a mammalian brain of experience-modifying connections in a way that was relevant to the synaptic basis of learning and memory, or believed to be relevant to the synaptic basis of learning and memory. These studies attracted the interest of people involved in neural network models, computational models, trying to understand how networks of neurons could modify their properties based on input through synaptic modification. And I thought, I'm an enormous fan of the Bienenstock, Cooper, and Monroe theory of synaptic plasticity, and a tribute to Leon Cooper, who died recently, who was the esteemed theoretical physicist for his work on superconductivity. But In neuroscience, he's also well recognized for developing this theory of synaptic plasticity. And what motivated him really was to understand the synaptic basis of learning and memory. But he tied his theory, or tried to anchor his theory, to experimental observations in visual cortex and the effects of manipulating visual experience early in life. And I should just say he was a mentor of mine. I trained with him, and I had a huge impact on my work. But to sort of get to the question that you originally asked, which is what other types of experience modify visual cortex and so on. Of course, our visual systems are modified by experience. That's how we're going to remember. We'll be able to recognize one another when we see each other in the hallway next week, right? We're forming visual recognition memories. That's happening in the visual system. But for years, it was thought that the primary visual cortex, the part that we've been discussing so far, is more machine-like in its function. And so that, you do some early programming work based on visual experience to get it tuned up, but then it's frozen in place, and then that information is distributed to the other 20 areas of cortex that are devoted to the analysis of the visual world, right? And that's been a prevailing view. I think that in recent years, it's become more and more apparent that there's still a lot of experience-dependent plasticity in the primary visual cortex. And I think that we probably need to be specific there to say what species are we talking about when we say that, right? Now, there are examples in the human visual system of experience-dependent plasticity that seems most likely to occur in primary visual cortex that serves things like perceptual learning. Why with training, for example, you can discriminate visual stimuli that otherwise you couldn't. So, with practice makes perfect, and there's some fine-tuning of connections in the primary visual cortex that serves that. But as you know...
What's like the paradigmatic example of that?
It would be, for example, I'm trying to think, there was a researcher whose name I do remember, named Avi Carney, who did some really important work on this. I think they were orientation discriminations, the ability to discriminate very fine distinctions in the orientation of line segments. And that could be learned. And one of the interesting things about that was that the learning would pop out after a night of sleep. So it interfaced with the role of sleep in plasticity. But, that's...
Is there a certain type of sleep that gives you that the learning?
Well, all sleep is good, but it appears that, it's such a wonderful question. And such an exciting area of neuroscience, honestly. We think about really two phases of sleep, two broad phases of sleep, slow wave sleep and REM sleep, rapid eye movement sleep. And our brains cycle through these different types of sleep in a good night of sleep. And each phase of sleep is thought to contribute something different to consolidating the synaptic modifications that occur during wakefulness. I'd say slow wave sleep, REM sleep is sort of sexy because it's considered to be dream sleep. We know we have wild experiences. More often than not, they're likely to be occurring during REM sleep. And REM sleep seems to play an important role maybe in like what's going on in adolescence, which is whittling connections down in REM sleep. Slow wave sleep has been a revolution in understanding what's going on in slow wave sleep in recent years. And one of the things that it seems to do is it cleanses the brain, which is important for all synaptic plasticity. But it also, there's a replay of experiences during wakefulness that occurs during parts of slow wave sleep that can consolidate the synaptic modifications. Yeah. But I wanted to get back to this question about experience-dependent plasticity in the context of learning. And I think in a way we have been fortunate, perhaps, that the world has been consumed with studying mouse vision. I laugh because mice have a terrible visual system. I mean, let's face it. They're nocturnal. They have laterally placed eyes. They have very little binocular vision. They have a rather poorly differentiated visual system. It's nothing like the beauty of a primate visual system. Nothing like it.
So who's the next in the chain after mice? Is it cats? Or?
Cats have been used historically, and they have a lot of features that are in common with the primate visual system. But the caveat there is that many of these features appear to be a consequence of parallel evolution, convergent evolution, so that it's not clear that, cats and primates diverge very early in mammalian evolution. So it may be that we may have all come from a rodent-like ancestor And cats developed the same solution that primates did, but we have to be a little bit careful of saying, oh, a cat's a small monkey, something like that. It's not that way. But there are other great, every species brings something interesting to the table. You have to take into account the niche that species occupies. So, let's face it, mice are nocturnal. They have laterally placed eyes. They don't have a lot of binocular vision. They don't really rely on vision. They rely on somatic sensation and the sense of touch through their whiskers and so on. But nevertheless, that's the species we've got. They're intensively studied because we can do genetic manipulations in mice. That's the main appeal of mice. They breed rapidly, have big litters, and we can do a lot of genetic manipulations in mice that have revealed a lot of the mechanisms of the plasticity that we're discussing today. But one of the features of the mouse visual system is that unlike a human, which may have, I don't know what the count is, but maybe 20 distinct different cortical areas that are devoted to the analysis of the visual world, mice have relatively few. So a lot of the functions that you might have to go searching high and low for in a human are also being accomplished in the primary visual cortex of a mouse. So there are types of learning relevant plasticity in the mouse visual cortex that are rather readily observed and easily studied. So the mouse has been great that way. I'll give you an example from our own work. One of our objectives here is to monitor the strength of responses from the two eyes, right, in visual cortex. That's the name of the game for us. There are a lot of ways you can do that. There's some very fancy, technically sophisticated ways, but there's some pretty primitive but very powerful ways as well. And one is a phenomenon called a visual evoked potential. So it's not recording the nerve impulses from individual neurons, but it's recording some summed output of a population of neurons. And the way to elicit a visual evoked potential is to show a stimulus that changes abruptly. And with every one of those abrupt changes, you average the signal that you're recording. And if you do that enough times, the average reveals a visual evoked potential. So it's riding on a very noisy background, but you just keep repeating this synchronous activation and you get a nice response. And that's kind of what we do in a lot of visual studies, is you do multiple trials and you average across trials, whatever you're measuring. The way we've been doing it in my lab and many labs is that we have the stimulus it's like a pattern of stripes. We call it a grading stimulus. So we have black and white stripes. And to elicit a response, we do a phase reversal so the white becomes black and black becomes white. So we phase reverse once, phase reverse twice, and keep doing that. You can phase reverse 100 times. And then you average the responses that are time locked to that phase reversal. And that's what gives you a visual evoked potential. So this is the way we monitor responses from the two eyes, right? Are you with me? Okay, cool. And we can do this through chronically implanted electrodes. We can put an electrode in the brain of the mouse, and we can plug them in and stimulate them every day. He's awake, looking at this computer screen. And a chance observation that a brilliant student of mine made, named Nate Sawtelle, who's now an esteemed professor at Columbia, he observed that over days, the response to that stimulus got bigger and bigger and bigger. At first, it could get twice as big.
Bigger, is this what do you measure? The potential?
Yeah.
So the amplitude gets...
The voltage deflection.
Voltage deflection, okay.
So it could go from 50 microvolts to 100 microvolts. So a huge change in the land of biology. And at first, we suspected it was a technical problem. What are we doing wrong? And we were building the plane as we were flying it anyway, because no one had ever done this type of recording in awake animals before. Nate was doing it. And Nate had the brilliant insight to say, well, I wonder if this is plasticity. And if it is, what would happen if I shifted the orientation of those stripes ever so slightly? So he plugged an animal in. After the animal had seen, say, vertical stripes for 4 or 5 days, the response had gotten big. And he shifted the stripes 45 degrees, and the response was right back down at the baseline. He described this phenomenon that we now call stimulus selective response plasticity. That, as one who's devoted his life to studying plasticity in the brain, I've never seen a more robust demonstration of plasticity than this one. No finesse is required. Any student can come in and we can get that experiment to
Did you
work.
try to do it for various
Yes,
degrees
yes.
and see how it...
Yes. And amazingly, as few as 5 degrees off of the experienced orientation, you'll see a significantly reduced response. So it's very tightly tuned to the stimulus orientation. So this is a gift to people that study plasticity. It's like, oh my God, we have this super robust change in the response to an experienced input. And of course, my instinct is to say, okay, let's try to understand the mechanism and do a lot of mechanistic experiments. And so we do that. We unpack it as much as we can. But of course, the deeper question, and the question that a reviewer invariably asks is, so what? What does this have to do with learning? I mean, this is a plasticity, yes, but what does it have to do with learning? Then I have to introduce another brilliant student of mine, postdoc, named Sam Cook, who's now on the faculty at King's College in London. And Sam had noticed that when the animals see the stimulus for the first time, they're awake, they would show this alerting response and they would show interest in the stimulus. But as the animal was plugged in on a daily basis to look at the same stimulus, they showed less and less of a behavioral response. And so they would habituate. It was a behavioral habituation. So first, and we could measure this. We could measure their forepaw movement, for example. So we saw this vigorous forepaw movement in the first trial. The days later, no movement at all. Then we shift the orientation and they start moving again. So the animals are behaviorally reporting that they recognize the difference in the orientation. And then Sam teamed up with, I have a whole bunch. I can't give credit to everybody because I have so many great people that are working on this problem. But he teamed up with another scientist named Rob Komorowski, and they did this in an open field. They showed that if they expose animals now in an arena with a computer monitor showing the stimulus, that initially the animals will run up to the computer monitor, they'll check it out. They're very cute, actually. They're really interested in the stimulus. But then next day, they're like, same old stimulus, nothing good happened, no interest whatsoever. And then on a test day, we can put the novel stimulus on and they show the interest again, right? So then we can plug them in and ask them, okay, well, are you responding differently to these experiences you had in this open arena? And yeah, so there's a big difference in the evoked response. So what we have is a behavioral report of learning, of actually a fundamentally important type of learning called visual recognition memory. And it's also learning to distinguish a familiar stimulus from a novel stimulus. And, a basic decision we have to make every day is whether a stimulus is novel and worth devoting attention to. It's novel because it could be a threat, or it could be an opportunity, or it could be nothing, right? And those are the 3 options. And so novelty detection is fundamentally important, not just to human beings, but to essentially every animal with a nervous system. And so to detect novelty, you have to have a way of encoding familiarity. It's just that simple. And then in the grand theme, it's visual recognition, how I recognize your face. So we now have this really great system. We have an electrophysiological readout of a behaviorally significant type of learning. And then you do the next level of experiments and you say, okay, do the mechanisms that serve this electrophysiological plasticity also serve the behavioral readout? Answer is yes, they do. So we, worked out. I had another great student I should mention is Misha Frenkel, who worked also with Nate in the early days to work out the mechanisms of this phenomenon. And then we were able to show that those mechanisms are essential in primary visual cortex for this form of memory. So we're getting closer. We feel like we're getting closer to us, the Holy Grail, of being able to point to the synapses that are actually encoding that type of memory. We're not quite there yet, but we get closer every day.
I'm just more curious from the optics point of view. Did you try to change the spatial frequencies and the brightness?
Yes.
So the visibility. I'm more inspired. Recently, Michael's baby was at our place, and somehow there was so many cool things around, but it would try. Yeah, the cats were running around, but then his kid would try to go to the display to
change the temperature on the thermostat.
And I don't know why for a kid it would be display is more interesting than something else happening in the room.
Right.
So I thought maybe it's the brightness, the acuity, the maybe spatial frequencies. Which affects his brain.
It's attention grabbing, right? What grabs your attention. But the things that attract attention or novelty, but of course, and motion, any moving stimulus attracts attention, and a stationary stimulus. Yeah, it depends. It's so great, actually. It's so cool, Misha, because it encodes orientation, it encodes spatial frequency, it encodes contrast. So all these features of the visual stimulation are being encoded as a familiar stimulus, and if I change any one of those features, it's seen as a novel stimulus.
Is there one thing, people seem to find like fractals more pleasing or interesting, or sometimes maybe in some ways less surprising because there's so many fractals in nature? That's one reason maybe why the thermostat is like more interesting than the cat, because the thermostat is not a fractal, whereas the cat actually is mostly a fractal. Like it's like a lot of fur, right? I wonder if there's like a fractal inside the brain that like compresses the information. If like you're looking at a tree, it's mostly the same thing over and over.
Exactly. Yeah. No, I don't know the answer, but it's a great thought. It's a great thought.
Did you try showing the mice fractals ever?
No, but I'm going to right after we finish. One thing we did do, one thing we have done is not necessarily related to fractals, but we've done sequences. So I have another former postdoc, Jeff Gavornik, who trained or exposed mice to sequences of stimuli, just different orientations of these bars, but always in a stereotype sequence. So I go A, B, C, D, A, B, C, D, A, B, C, D over time. Yeah. So the animal will just look at A, B, C, D, and then come in the next day, look at A, B, C, D. And then on the last day, he would scramble the order and see what's going on. And so in some ways you go, okay, well, yeah, of course, it's going to show a phenomenon not like the stimulus selective response plasticity, what we call in the lab SERP, SRP, SERP. But in fact, it turns out to be different from SERP, not necessarily more interesting, but different. And the key experiment, the one that really blew me away, was that after the animal's been trained on A, B, C, D, and remember, we're recording the electrophysiological response, A, B, C, D, right? He shows stimulus A, and he omits stimulus B, and we still see a signal in the brain that is the signature of the occurrence of stimulus B. In other words, I don't know what the animal perceives, but the brain is behaving as if stimulus B is there because it was anticipated by stimulus A. And if he takes another orientation that the animal hadn't learned in sequence, you don't get that, what we call phantom response. So the animal, this is in primary visual cortex. So there's a deeper level of plasticity that's keeping track of time, and the timing of those stimuli mattered. Right. So if he altered the time, he also didn't see the effect. It's a playground, honestly, to do clever experiments in primary visual cortex. And these are all, the experiments that I've described so far, are just unrewarded stimuli. We haven't gotten into fancier things like adding reward, which will engage yet other circuits. But this is just passive stimulation.
This is truly interesting research. And you mentioned visual memory. And what at this point we know about different types of memory? It can be different sensual memory. It can be other types of memory.
Well, that's a vast topic. There are, there's a useful distinction that is made between what's called declarative memory and procedural memory, or non-declarative memory. And to give you an example, declarative memory is usually what we associate with the term colloquially. It's a memory for facts, events, places, and faces. Right. The memory of our conversation today is a declarative memory. And it's in part because I can declare that it happened. There are also types of memory called procedural memory. And these are memories that usually, declarative memory, one of the features is they're rather easily formed. Right. We remember the beginning of this conversation where you were setting up this podcast thing. They don't require practice. We're streaming this information in right now, and it's being stored. Procedural memory usually requires repetition. And it's the kind of memory that you rely on to learn new skills. And so these two types of memory are served by different parts of the brain and different mechanisms in the brain. So there's some very famous classic textbook examples of impaired declarative memory. For example, a very famous patient named patient HM, who had damage to the temporal lobes that cause amnesia. So HM, if he had come, we had never met him before, he had walked in here, we'd have had a nice conversation, but he'd have no memory of the conversation whatsoever. If he left to use the men's room and came back, he would not recognize us or having ever seen us or had the conversation. And yet, so HM had a very devastating amnesia, and yet it was shown that HM could still, with practice, perform new motor skills. So memories were being laid down, but it were involving a different type of memory, a different type of circuits. And even in the context of visual memory, I'll give you an interesting example. I go back to our own work on SERP and sequence learning, but I mentioned HM. This truly is a very famous case, and the damage was to the hippocampus in the temporal lobes. And this is a structure that's very intimately involved in memory storage. Now, this phenomenon that I described called SERP, stimulus selective response plasticity, does not require the hippocampus. We can take the hippocampus out. It's fine. No problem. Okay. So it's a low-level type of memory. The sequence memory, on the other hand, does require the hippocampus. So there's a circuit that's engaged by these sequences that is involving participation of the hippocampus that is necessary for those memories to be formed. So again, I think we have an opportunity. I'm not going to live long enough to work out these. I see my mortality, but we're setting the table for you all to go solve these problems because, it's been known for a long time that, hippocampus is important for declarative memories, but there is not a satisfying answer to precisely how, particularly those that are stored in the neocortex, like visual cortex.
Also interesting on the hippocampus, I was reading about it related to the lectures. It seems like it doesn't have, it's considered to be the main part responsible for memory, and then it contains only something like 10 million or 15 million neurons. So how come the memory only uses that few neurons?
Well, I think that the key point here is that, I'm glad I brought up HM. So this patient, as I mentioned, has this devastating amnesia, so-called anterograde amnesia. So from the moment that the hippocampus was removed, he's incapable of forming any new declarative memories. However, the memories that were formed years before the hippocampus was removed are intact. They're just fine. So the hippocampus, it's storing information, but it's storing it temporarily. So you don't have to have a massive memory store. In a way, it's a buffering memory.
So it's more like RAM if we compare it to the computers? Or?
I don't know if RAM would be the right comparison because RAM goes away if you turn the computer off, right? So the hippocampus survives,
maybe short term, or comparing to hard drive where it can stay much longer over a period of time of life.
Yeah, that sounds about right to me. I think that a lot of the work in the hippocampus literature gets confusing fast because if you approach it from the perspective of, say, HM or visual recognition memory, it seems you can tell a story of the hippocampus and how it's involved, even though we don't know precisely how, but in general concept. However, another way of approaching the hippocampus is through work in rodents. And one of the major challenges that rodents face is navigation, right? So they have to navigate through tunnels, and you have to go, when they go foraging at a great distance, they have to find their way back to the nest. And in rodents, the hippocampus is deeply involved in storing that memory. I fear, and again, my hippocampologist friends are probably going to be giving me a hard time for this, but I think we have to be very, very careful about overgeneralizing what is observed in a rodent to how we use our hippocampi. We humans use our hippocampi. And I should say, you say it's small. It is small. In a human brain, it's very diminutive. If you look at in a rodent, it's huge. It's a massive structure relative to the rest of the cortex. You look at a human, it's like, you got to dig for it. It's down there buried in the deep reaches of the temporal lobes. So by size, our massive capacity of learning information and combining in interesting ways, that's got to be in the neocortex, the rest of the brain. It's just the hippocampus is like this bottleneck, really. And that if you constrict the bottleneck, then you can't lay down the tracks of new memories. But the memories themselves are outside of the hippocampus.
Is there a problem of running out of memory? Or the people when they...
I would say the answer is definitely yes, from personal experience. It depends on how many neurons you've got.
Is it the limitation of number of neurons or something else, maybe?
I do think that the limit is set on the number of synapses. And, one of the revealing features of neural network models. Yep, like we have this brain filled with these unreliable cells called neurons. They're quite unreliable. Their firing is all over the place. They goes up and down with levels of alertness. We can't generate any new ones except for in some very special cases. So the neurons you've got now, I always tell my classes, you have as many neurons today as you're going to have, because tomorrow you'll have a fewer, because they do die. So we have these unreliable elements, and yet we can form these robust memories. And what saves the day is simply the number of neurons and the trillions of synapses. So the memories are formed in this pattern of synaptic change that can really tolerate the loss of individual elements in the neural network. But that is not to say that it won't degrade as you start to pluck out neurons. And the neural network models yield this property called graceful degradation of memory, so that as neurons are removed, instead of there being catastrophic loss of a particular memory, like poof, there goes grandmother, what you start to see is what's called graceful degradation, where you start to maybe confuse memories. If you have an elderly family member, this is what you typically see, is that they start to get confused. They may see you first as a child, and then as a son, and then as the youngest son, and they work their way through it, right? But it's not at the snap of a finger, right?
I guess I have like two related questions. One is, if you could grow additional neurons in a dish, the way they do with, for example, for cancer, they do CAR T. If you could grow a chunk of neurons and put them in your brain, what's the realistic chance that they'll integrate into the system and start doing something relevant?
Good question. Actually, before I answer, I want to say that there is some adult neurogenesis in the brain. The olfactory epithelial cells that are part of the central nervous system, they will regenerate. And also a part of the hippocampus called the dentate gyrus has neurogenesis. But it's quite modest. But there's some evidence that some drug treatments can promote neurogenesis in the hippocampus. And usually this is discussed in the context of regulating, mental illnesses. So setting that aside, the question you're asking is, can we transplant in more neurons, basically? And this has been a dream. And the challenge is integrating, because, as we were discussing earlier, brain function requires really precise wiring. And reestablishing that wiring is challenging. You can put the neurons in, they can stay alive, but they don't integrate well. So it may not be.
How can you tell if they're integrated or not?
A lot of different ways, all the tools that we have available, anatomical, electrophysiological, and behavioral, to see whether there's a recovery of function. The best path to recovery of function is to make the most of the neurons that you still have, actually. But there are some cases where there's some functions that don't require that level of precise wiring. And I'll give you an example. It would be the motor disorder called Parkinson's disease, which is a primary cause of Parkinson's disease, is the steady loss of neurons that synthesize dopamine. Neurons in a part of the brain called substantia nigra that synthesize dopamine. So dopamine is like a lubricant on the circuits that control voluntary movement. And as these neurons die, you start to lose that lubricant, and the machine starts to freeze, and you can no longer initiate voluntary movement. And the medical strategy to treat Parkinson's disease, and the one that every patient that has Parkinson's disease is treated with, is to try to boost the production of dopamine. So you give a metabolic precursor to dopamine called L-DOPA, and that can cause those remaining surviving neurons to produce more dopamine. And therefore, you get more years of useful life. You're not stopping the decline of those neurons. They're going away, but you can better utilize the ones you've got. But it seemed like this would be a great opportunity for a transplant strategy, to put in dopaminergic neurons, even neurons that are engineered to secrete dopamine. And honestly, I haven't been following the field closely in the last few years, so there's probably been more progress than I'm aware of. But they were encountering the same problems of integration, of getting those dopaminergic neurons to secrete the dopamine where it was needed, which is in a part of the brain called the striatum. Again, I haven't been following the field, so I would hope that they've made more progress. But that's going to be the low-hanging fruit.
Is there a world where you could take like an eyeball and a brain chunk and use it as like to drive a car, for example? Like connect it to some electrodes that control the foot pedals and the steering and drive a car? Like, is there something fundamental preventing that, or what?
No, that world, sort of that world is we're on the threshold of that world in the sense that the relationship between visual perception and guiding a motor action, right? So that's an interface right there between vision and action. And people that suffer from spinal cord injury, they paraplegic, quadriplegic, that's been disconnected. They can still see, they can still visual perceive, but they can't drive the car anymore because they can't control the movement. So that's where these neural interfaces come in, brain-machine interfaces. What they really rely on actually is the very nature of the coding that's used by the nervous system. Remember, we're talking, we have unreliable neural elements. We rely on trillions of synapses and some sort of summed output, right? The individual neuron vote is very minimal, but it's the mass action, the many neurons active at the same time that are actually encoding information or representing information. And so it's in a way, the crudeness of that output that gives us opportunity to develop these brain-machine interfaces, so that the earliest attempts were done, for example, what we showed like in the class of a brain gate was developed by my colleague when I was at Brown, where they have an array of electrodes, I don't know how many, maybe 64 electrodes that they can put in over the motor part of the cortex, the part of the cortex that issues the commands to do a voluntary movement. There are millions of neurons in that part of the brain, right? They got 64 electrodes. Like, how much useful information you can get out of 64 electrodes? Well, it turns out quite a bit of useful information. So what they would do is listen in to those electrodes when they would ask the patients to visualize the motion, the movement, not visualize in the vision sense, but to imagine themselves making the movement. And they would record that activity. And then what they would do is try to, and now with AI, this has probably gotten much more straightforward, is to then decode that pattern of activity. So to relate the pattern of activity to the action, the desired action. And then they could move, robotic arms or cursors on a computer screen. So that's kind of where that's at right now.
Sounds like science fiction,
It does. It sounds amazing.
but it is amazing, truly amazing. It's happening right now.
Also, from engineering perspective, like you measured, it was only 64 electrodes, and I guess what you also do with the mice experiments with the stripe patterns. What is the direction? Do you try to increase, like, get this like a matrix of the electrodes, increase the density, or I don't know, maybe use like other ways of exciting and measuring those neurons' activity?
Yeah, I mean, I think that you're absolutely right. Your intuition is exactly correct, which is the more data that comes in, the more straightforward it will be to do a fine graded encoding or decoding of what that activity is representing, right? So, now it's very routine for us to record from a single electrode, 64 channels, 32 channels of information, and kind of arrays that could be quite vast. So technology is definitely advanced to the point where you get a lot more data. And the challenge is how you're going to handle that data and make sense of it. And that's where all these great computational tools are coming into the equation.
Yeah, with this like fascinating, incredible examples of BrainGate and also what now Neuralink is doing,
Yes.
like similar things but wirelessly. Where do you see like from the technology point of view, or like maybe the applications, what would be like the next milestones in this path?
I'm probably the last person to ask that question too. But from my perspective, I think achieving the objective of improving the quality of life for people that have lost voluntary movement, that's the objective. And we know in principle it can be done. And it's a question of just reducing it to practice in a way that's available to people that need it. It's not an exotic technology. It's actually a technology that's available. And I think that's really exciting. The other way at this will be to come up with new therapies to promote regeneration of the spinal cord and reestablish the connections in the brain or in the central nervous system. There's been some steady progress there too. It's obviously a big challenge. People have been on this for a long time, but you're starting to see a lot of progress there. I would suspect that in your lifetime, that'll be the standard of care for treating people with spinal cord injury will be substantially improved from what it is today.
I guess I can maybe slightly change the question. If you have unlimited funding, what kind of project would you do?
Me personally?
Yeah, unlimited resources,
Oh,
people,
Lord.
funding, financial, anything.
Oh, God. Oh, yeah, that would be great. Let me just dream of that for a second. Well, I think that every scientist you pose that question to may approach it differently depending on where they are at in their lives and what paths that they've traveled. And so I just give you my perspective. And it may seem like my view is very narrow and parochial, but I have mixed feelings about my experience in neuroscience over the last several decades. On the one hand, the advances are astonishing, like especially the technical advances. Amazing. Almost any experiment you can conceive of can be performed now. I mean, it's really amazing to me, but it's sort of the contrast is in a way, despite that amazing progress, how little we've actually accomplished. Like some of the fundamental questions that were posed 50 years ago, it doesn't feel like we've made that much progress on. So each one of us that works in an area, I mean, I'll give you my own personal story. So I work on the fundamentals of experience-dependent plasticity in the brain. We've talked about, I use the visual cortex for well-considered reasons. We've made a lot of great discoveries. We're really excited about it. Over the course of that time, you start to see how the work that you've done can be applied. So it's not, we're basic scientists. We don't wake up in the morning going, what disease am I going to correct today? Rather, the opportunities present themselves. So I talked about monocular deprivation. The human clinical condition is called amblyopia. There are millions and millions of people that suffer from amblyopia. It can have a very negative effect on their quality of life. And so a question that we can ask is, can we leverage what we've learned about synaptic plasticity to develop novel treatments for amblyopia? And I would say the answer is definitely yes, right? And we are really limited. We're resource limited, right? We have some great wild ideas. And if somebody came in and rained money on me, we could pursue that in a much more aggressive way. We suffer. I mean, how should I put this? We are extremely grateful to the support that we get from, granting agencies, the NIH and so on. But these dollars are very, it's very competitive to get them. They're never enough. They're also funding work that is more conservative than what you'd really like to do. Essentially, the sad truth is that you almost have to have completed the study to convince the reviewers that the study can be completed, right? We're not liberated to do the science that we think could make a big impact. We just typically are not. And the other disease area that I work on is Fragile X syndrome, which we may not get to, but what's a devastating neurodevelopmental brain disorder. We've seen our way to developing treatments for Fragile X syndrome that we know can improve the quality of life for these families, and yet we're resource limited. We can't get from where we are today to where we need to be. So that's what I would do. And again, look, I'm at a certain age now where I know, considering how little progress we have made in helping these people over the last 40 years of my career, I got maybe 20 years left, right? So I have to say, okay, what am I going to do in my time left to reduce this to practice in a way that has a lasting legacy in addition to the knowledge that we've created, which, I think is fine. Great. And so those are the things that I would spend my money on if Lord showered me with money, is I would develop a treatment for Fragile X syndrome, and I would develop a treatment to cure amblyopia in adults. That's what I would do.
What kind of treatment would that be for Fragile X?
Fragile X. So that's a whole nother story that we haven't had much chance to talk about, but we can talk about it now. Again, a scientific career is really wonderful because you never know, when you keep turning rocks over to see where they're going to lead you, and you never know where it's going to. Much of my early career was devoted to understanding the mechanisms by which visual deprivation makes synapses weaker. Okay, that was where we began. And in the course of studying that, we found a form of synaptic weakening that required new protein synthesis at synapses. And we don't have to go into the excruciating details, but that was the novel discovery. We didn't know anything about protein synthesis regulation. The question pushes you into areas that where you feel like an imposter. It's like, oh God, I have to learn about protein synthesis regulation. Well, it was that push that we became interested in this protein called FMRP that we had suspicion played a role in this type of plasticity. Of course, how do you study that role? Well, you get the FMRP knockout mouse and you see what's different about it. Well, it turns out that FMRP is the one single protein that is missing in Fragile X syndrome. So Fragile X syndrome, just a quick review, it's a monogenic disorder. It's caused by silencing of one gene that encodes this one protein, FMRP. The consequences are intellectual disability, autism, irritable and aggressive behavior during childhood. So there's a lot of increased incidence of childhood epilepsy. It's a terrible disease to have. So we're studying this process, and we found that it was altered in a way that we had not anticipated. So it was like big surprise. And this discovery led to suddenly the eureka moment was the recognition that we might have a way of developing a treatment that could not cure Fragile X, because to cure the Fragile X requires re-expressing the gene, but to address many of the seemingly unconnected symptoms of the. So we would call that a disease-modifying treatment because it can improve multiple aspects of the disease. This treatment approach was validated in animal models. So we and now many labs around the world have developed treatments that substantially improve the course of Fragile X syndrome in mice and rats and fruit flies and zebrafish. We're teed up for a success in human beings. The problem is that to go from the bench to the bedside is extraordinarily expensive. And the way that that gap is filled in the system in which we live is usually private investment, venture capital, things like that. And the stakes of the game are huge, right? So we go from maybe hundreds of thousands to low millions of basic science research to tens of millions to hundreds of millions to go into a human clinical study and see it all the way through to the end. Unlike experiments where if at first you don't succeed, you can try again, right? It's just you get some more mice, right? You change the experimental conditions a little bit and you just keep chipping away at it till something works. In human studies, it's like betting the house on every trial that you do, you make your best educated guesses, but nobody's perfect. Some people get it lucky the first time, and those are the ones that think they're brilliant, but frankly, they got lucky, in my opinion. But you try, and then the trick is more often than not, you won't be successful on a first try. It's iterative process. You refine the molecule, you establish the target, you find the dose range that works, you find the endpoints that are critical to measure a benefit. You convince the FDA that this is improving the quality of life for a patient in a trial that's in weeks in duration, but you're trying to change a disorder that is a developmental brain disorder, right? That emerges over years of life. So there's a lot of these sorts of obstacles that you have. And if it's a rare disease like Fragile X, you don't get too many shots on goal. And the reason is because it's an investment thesis. Those dollars are being invested in a treatment that the investors are expecting a return on. In venture capital world, they're used to lots of failures. They'll invest in 10 different projects in the hopes that one of them is $1 billion maker. But you don't get more than 1 or 2 chances, or they just go away. They just move on. They go, our dollars are better spent somewhere else. So they don't have the commitment to the disease. Whereas, if you sit in my chair, I'm committed to the disease. I want to give it the best shot and develop a treatment. I have a great treatment for Fragile X, but what I'm lacking is the tens of millions of dollars are going to be required to make it happen. So yeah, so if the skies opened up and money poured on me, if I win the lottery, I promise you I will spend it on this. Okay. And occasionally I do play the lottery.
Wonder how many other diseases already can be cured in the same manner.
Yeah.
And just didn't get funding yet.
Most probably. Yeah. Especially rare diseases. I say the struggle in a Fragile X, which is considered a rare disease, it affects 1 in maybe 4,000 people. But if you have an indication like generalized anxiety disorder or even Alzheimer's disease, which is going to affect, then you get a lot more shots at goal. And why? Because the upside is that much larger. So in Fragile X, you may venture $20 million. In Alzheimer's disease, you could venture $1 billion because the payout's going to be many multiples of that. So it's just the way the system works. It's capitalism for better or worse,
Yeah, I mean, the kind of game theoretic solution to this, which maybe is beyond the government's capability is obviously to make the threshold lower for rare diseases. But I don't know, maybe they do try to do that, but in effect, it's still very high. Or just make the studies easier to run, like these kinds of things, because obviously there's probably no financial sense in actually investing the same amount in the smaller diseases versus like the Alzheimer's. And also the risk is way lower for rare diseases because the number of people you affect, let's say you do have a drug that gets through and gets approved that's ineffective or dangerous, the number of people you affect is much lower. Like if you approve a terrible drug,
Oh,
which
right.
still happens, anyway,
Sure.
but if you approve a dangerous drug that's given to 50 million people or whatever, the impact on society is huge. So, like, it kind of makes sense to have those high thresholds. But I guess the tragedy is that they don't know how to do that. They don't have a knob that turns, smoothly
Yeah.
to
I mean, you're
like,
totally, you're exactly right. And there are a couple of knobs. I mean, there is something called orphan product designation, which gives you a leg up if you're targeting a rare disease. For instance, one of the legs up is if you were to successfully develop a treatment for which there was no patent life, you would still have, I forget the number, 7 years of what's called market exclusivity. So you could market into that to recoup your investment. So there are a few incentives in place so that these rare diseases aren't neglected, and that's great. That's great. I think that one of the challenges in the regulatory domain is convincing the FDA. The FDA's role is to ensure that, first and foremost, that any treatment that's developed is safe. Safety first. The question of efficacy gets a little bit like a gray zone where, yes, you want the data to support efficacy for sure before it's marketable. But again, it's going from a mouse to a human. You got to make so many guesses as to how am I going to measure the benefit and what's a reliable measure? How are you going to avoid placebo effects? There are so many tricks that have to be put into play. And then ultimately, I would just say patients and caregivers are often denied the opportunity to try a medicine because that bar is just too high. And that doesn't seem right to me. I think that they should be given the opportunity to try, and then they can do a post hoc analysis of whether the treatment's lived up to its billing. If it's safe, safety first. Right?
what's your recommendation for younger people that are getting into either neuroscience or just getting into academia, and like how to have a enjoyable career, find interesting things, and also teach new students? Do you have any advice for younger people getting into this field?
The main advice is difficult to find the right word. I mean, I like to say that you have to love what you do, you have to like what you do, you have to have a passion for what you're doing, and it doesn't matter what it is, any walk of life. And that's the key to happiness, right? It is to do what you love to do and feel passionate about. And I think a scientific career, there are a lot of obstacles. It's hard. It's hard. Science is hard. Experiments don't work. It's hard to get funding. There's a lot of obstacles to a science career. But you can't let those obstacles obscure the fact that you're in it for the joy of discovery. And, there's no greater joy, if you will, of revealing something about nature that had never been known before. It's just an incredible experience, exhilarating, right? And how often in life do you get to be exhilarated? So I think it's a wonderful pursuit, science, and don't get hung up on being too overly technical. Oh, I got to do this and I got to do that. I got to check off all these boxes to be successful. It's like, well, that's a good idea. Be smart about it, be adaptive, but don't lose sight of the fact that you're in it for the joy of doing the science. And then as far as teaching goes, I started out as a teaching assistant in the course, not so different than the one I'm teaching you guys. First of all, it's enjoyable, right? You can just see the students' eyes light up when you finally get a concept that you've been trying to get them. And so that's rewarding, right? But the thing about teaching that maybe is unexpected is that it forces you as a scientist, it forces you to survey a field, to render it down, to identify what are the key concepts and principles. There's so much information out there, and it's terrible now. The proliferation of data and papers is like obscene. But the challenge is to say, how do I capture the critical concepts of the field and teach those? And how do I identify the big problems and set aside the smaller problems? And I think that as a scientist, you benefit from teaching because it helps you to keep your eye on the big problem and set aside the smaller problems. And so your science will be more impactful. because you're imagining, I mean, here, the dream of every academic scientist is to one day have it taught in their introductory physics class or neuroscience class. It's like, yeah, man, I finally did it. I added a brick to the foundation of the field. And you try to picture your work as like, why is that worthy of being taught to an undergraduate in intro, whatever, right? So did that answer your question?
Yeah. I guess I have a bonus question, but you don't have to answer this because I don't know if you've ever thought about this, but if you look at the original neural network, like AI, it's kind of inspired a little bit by the visual system, right? So they're looking at layers and activations between those layers, just very vaguely inspired, right? Is there knowing what you know about the systems around plasticity and learning, have you thought about, okay, if I had to turn this into simple rules that could be programmed into a computer to build some kind of memory system or some kind of neural network memory, what would that algorithm be? Have you thought about some kind of algorithmic mapping what you know about the biochemistry of the brain and the connections in the brain to like an algorithm?
Yeah, I think, that's a little above my pay scale, whatever. But I will say that, I was very influenced by working with Leon Cooper. I mentioned Leon before. He's the C of the BCS theory of superconductivity. And he developed this ridiculously simple yet extraordinarily powerful learning rule. And it's really guided a lot of the experimental work that I've been a part of. But it's also attracted the interest of people doing AI and neural networks. In fact, it was originally designed to be implemented in neural network models to solve real world problems like visual recognition. And Leon and I published a number of papers together over the years. And I did one collaboration with someone here at MIT where they were putting this theoretical learning rule on a chip. What amazes me is I see all these citations, papers on which I'm an author on topics that are way, in electroengineering journals and things. So it is being done. It is being done. These are biologically inspired learning rules that are actually being implemented and hopefully add something to what's possible now, maybe in terms of energy efficiency.
Yeah, that's really interesting.